Hey, hello everyone. Good evening. Uh, hope all of you can see my screen and hope all of you can hear me. Can I have a quick confirmation, guys? Can someone please confirm? Are you able to hear me?
Yes,
Yeah,
sir.
it's good.
Hi, sir.
Yes. It is
Hi.
visible.
Perfect. Thanks for the confirmation, guys. All right. So I don't know how many of you are already part of something related to clinical or something related to pharmaceutical. I'm not sure of that, but I'll be under assumption that all of you are newbies to this particular technology. I cannot call it as a technology, I can call it as a domain. So I'm going to be your trainer for clinical SAS training. All right. So just to add, clinical SAS is nothing but a basic SAS technology. I cannot call it as basic exactly. I can say that a SAS technology along with all the clinical standards, right? Here, we need to understand that you have already completed the SAS part. That is what my assumption is, right? I can see some of the people who have already learned SAS from me. I could see Tejashwini, Mohanty, who all joined here. So I know these folks who have already completed the SAS training. I'm also under the assumption that others also have completed your basic SAS, I mean to say how SAS works, what are the manipulations we can do there, how you can do the ETL, how do you do the reporting, and what is the basic analysis, right? So I'll be under assumption that you have already completed that. And in clinical SAS, we don't discuss anything or we don't what to say, teach anything related to the SAS syntax or something related to SAS procedures. No, I don't deal any of that. Instead, I'll be concentrating most of my time on explaining the clinical standards, or I can say that all the clinical domain related stuff. There are so many domains we're going to discuss one after the other. And while learning these different domains, we are going to use SAS skills. We are going to use SAS skills to implement few of the things. So how it is going to be? We are going to start learning about all the clinical domain knowledge. The main purpose is to acquire the clinical domain, right? Along with that, we are going to use our SAS technology skills to implement few of them. This is how we are going to proceed, right? So it is going to be most of the time we will be discussing about the clinical standards. Right. Today, I'm not going to touch any of this. I mean to say that what are the various domains we have, what are the other stuff. I don't touch any of that. Today, strictly I talk about the very basic or the core structure of this pharmaceutical thing, the core structure of this clinical thing. I'm going to talk very basic. I mean to say, I consider all of you are the beginners in this particular domain. So I'll start from the very basic. So without wasting much time, let's start discussing about our very first topic of the day, which is nothing but drug life cycle. Maybe even before that, we also need to understand about what is called clinical trials. Most of the time we use a term called clinical trials. So what is a clinical trial, how and why it is important, why there is so much of research around it, what are the outcomes of this particular clinical trials, to implement these clinical trials, what and all required, and finally we discuss about where SAS comes to the picture, right? Because we are talking about clinical SAS, right? Why SAS is required in the clinical trials. So all these things we are going to discuss in the today's session. All right. So with that background, I'm going to start my discussion. I'm not a very big fan of using the PPTs and all, but since it is going to be more theoretical in nature, today's session I'm going to use some of the PPTs. All right. So before I proceed with anything else, I just need a confirmation from you. Are you clear with what we are going to discuss or what is going to be the agenda for today? All of you are comfortable with that? Or any questions? Feel free to ask me. All right.
It is okay, sir.
Perfect. All right. So let's start with our very basic discussion about what is this clinical trial. We are going to use some of the real world scenarios to understand this particular piece. All right. Okay. First things first. So when we talk about the clinical trials, the very first thing we need to understand is because without a medicine, we cannot live. That is the current scenario, correct? Irrespective of how healthy you are, irrespective of how strong family background you have in terms of your medical history and all, though you are coming from a very good solid background where you don't have any of these hereditary diseases or and all, but still medical medicine is the most basic thing, basic need which is in the current scenario, right? It is very, very basic. I'm not talking about the medicines which is going to cure us. I'm also talking about the medicines which are going to prevent the diseases. I'm also talking about the different equipments which are using on our day-to-day basis. All right. So we talk about before we take any medicine, there are some of the treatments which that particular drug will undergo, right? So any medical drug you take, anything, right? Or you consider any medical treatment, or you consider any preventive drug that you are talking about, or any medical devices that we are using for our safety, or which are we are using for the diagnosis, they will go through a specific cycle. They will go through a specific process, and those cycle or those processes we can call it as clinical trials. What is that? I'm not talking about just the medicine or just the drug that we are taking. I'm talking about a broader picture. The picture that I'm talking is that can be a medicine which will cure the disease, or that can be a medicine that is going to prevent the disease, or we are talking about some of the devices which we will use for diagnosing our all these together, irrespective of whatever it falls under, whatever the category it falls under, it will go through a specific cycle, and that cycle we can call it as a clinical trial, or even you can call it as a process, right? So to be specific, what exactly it deals with? Mostly, I understand that it is going to be a clinical trial. Okay, fine. So they will go through sort of certain procedures. What are them actually? On a very high level, I'm talking about at a very high level, right? Clinical trials is mostly about research studies, deals with either medical or health related stuff. There is a minor or thin line difference between these two, right? So there are going to be medical research studies and there are going to be health related studies, research studies, because most of the things falls under here are, right? So if you carry out those trials on the human beings, you can call them as the clinical trials. Similar kind of a trials we can also do on the animal. And if you do that, we can call it as a veterinary study, right? But we are not interested to go with the second piece for now. We mostly concentrate, or not, I cannot use the word mostly here. I can say that 100% we will be concentrating on the first one, which is nothing but the research studies that we are concentrating on medical or health related stuff, we can call it as the clinical trials. Guys, I understand that some of you might have clinical knowledge, prior clinical knowledge. For you, it might be a bit boring. Today's class might be boring for you because you already know all this. If you are not aware of this, but still if you are part of the clinical process, well and good. But if you are already in the industry for some time, and if you already have a very good detailed understanding about what I'm talking about here, maybe you can just take it as a breezy note, on a very light note you can consider. All right. Otherwise, you may drop also. Today's class is primarily concentrating on the clinical trials. Hope I'm clear with that. Because some of you may feel that, hey, I'm already aware of all this. I know all this already. Why you're going with this? Because you might aware of this, but the people who have joined along with you may not be aware of it. That's the reason we are doing it. All right. So just to start, I started talking about what is called clinical trial. So a clinical trial is nothing but it is going to be a research or a medical research, or it is going to be a health related research that happens on the or that carry on the humans. The same research, if I do it on the animals, we can call it as a veterinary. Right? So that is the first part. Now, just go with why it is important. What happens if I don't have a clinical trials? Correct? What happens? If I don't use them, there are a lot of definitions around it, why it is important at all. But in a simple way, if I want to talk about it, only two factors which are important to me. The two factors which are important to me are how safe the medicine is. The first thing is going to be how safe the medicine is going to be, right? I can have hundreds of medicines that are available in the market, correct? But some of the medicine we will take, some of the medicine will take strictly under the physician guidance, the general physician guidance. Some of the medicines, especially in India and all, we can even just buy it from our local medical shop and we can have it. But few, or definitely we need to have under the some general physician supervision. Otherwise, we cannot have it. Why? Because safety. If you have a normal paracetamol, anyone can have it. There is no problem at all. But when it comes to the medicines, few of the medicines, until and unless you have a proper supervision, you cannot have it. So the main goal or the main objective of carrying the clinical research is to ensure the safety of the drug. Number two, along with the safety, I also need to talk about the efficacy, or we can simply call it as efficiency. Maybe we can go with the simple general terms. We also need to talk about the efficiency. All right. So why efficiency is important? Why efficiency is important? Um, there is a debate always going on, but on a lighter note, I'll take this. I'm not criticizing anyone or I'm not supporting anything here, but generally I'm talking. There is an Ayurvedic medicine and there is an English medicine, right? Both of them are going to cure the respective disease. Let's say that. Most of us will be more inclined toward the English medicine because the results are quick. I can simply say that those are more efficient compared to the other Ayurvedic medicine, correct? Because both of them are going to cure the disease, but we need highly efficient drug. The one which is going to provide the quicker results, the one which is going to provide the better result, at the same time, the one which is supposed to be safe. So the main objective of the clinical trial is to provide or to ensure these two factors. What are them? The drug must be safe enough. Number two, the drug must have the very high efficiency. Is that clear? Right? A very simple stuff. This and all, most of these things are well known to us, but still, we're just having the baby steps towards the clinical science journey. All right. So what is the scientific method? Because if you see this, the previous slide, the first one I haven't discussed about, all I just carried on the second one, because why it is important, I concentrate on that. But prior to that, in clinical trials, on a lighter note, the researchers, the R&D department, research and development department, they take all the results from the basic scientific research and then they translate them into the different ways to prevent or to treat or to diagnose the disease, right? Let's say if you take the vaccines like, the corona vaccine, COVID-19 vaccines, right? All these are vaccines. Why we are calling them vaccines? These we are taking to prevent it, just to avoid fall under your COVID, just to avoid that particular one, correct? That is a preventive one. So let's say that you already have fever, so you're suffering from fever. So you're going to take some paracetamol. The drug here is going to cure, to treat your the disease that you're already suffering with, right? And there are few which can be used for the diagnosis purpose, right? So there are some eye drops which will be used to dilate your eye. The primary reason of dilating your eye is going to be to see how good your retina is, to see what are the issues that are having, various issues that are having with your retina. To go with this particular retina test, your eye need to be dilated. For that, you need to use the diagnosing basis. Correct? The eye drops that I'm going to use, not to cure, not to prevent, but this is used to diagnose my eye. So there are three different types we have here. One is to prevent, second is to treat, third is to diagnose. But to produce all these ways, or to translate my scientific research into these three ways, I'm using the clinical trials. But that leads us to a question, and that question is going to be, what is called this basic scientific research? That one we are going to discuss here. So the right hand side diagram is pretty much self-explanatory. So that is going to give you the answer for what exactly is the scientific method is, right? So clinical trials are nothing but the real world applications of the scientific method. Let me talk about what exactly this is. Let's say consider any drug, whatever the drug it is, all right? Or consider any medical equipment. Maybe you can consider your, the glucometer, which I can use it for testing my, glucose levels, or you can take your BP machine, or you can just use your, the device which I can use to check my temperature. All these are various devices, correct? So these, any of this drug or any of this supporting device, first it will get tested. The moment the test is complete, we raise a question. Okay. So I just use this particular one to check the temperature. The question that I'm going to ask is this the correct temperature or not? I need to cross verify that. So a question is going to be raised. Then followed by a hypothesis is made. Mean to say that, hey, I'm getting this temperature under the normal room temperature. Correct? When you are within the room, you are checking your body temperature by using your whatever the equipment, either it can be a sensor one or it can be a holding one, whatever it is. I'm using it just to test the temperature. What if I'm in a extremely different conditions? I mean to say, I just switch on my AC. I'm under 16 degrees temperature. My room temperature has come down to 16 degrees. What if I use the same device to test my temperature? Will I get the same result or not? Right? A hypothesis is going to be. And then again, we are going to conduct an experiment. Because I made a hypothesis, I have asked a question. To fulfill these two, I need to experiment again. I'm going to get the result, new result. That new result we are going to analyze again. We do this multiple times. So what we are doing, I have an equipment. I will give the equipment to you. You started experimenting that. So what you are going to do, every time I give a device to you, first thing is you are going to test it. Then you are going to raise a question. Then you are going to make a hypothesis. To fulfill your hypothesis and to answer your question, you are going to do a retest on that particular one to meet these two requirements. The results are going to be generated. Analyze the result and get a conclusion. This conclusion, you can get it in the second test, or you may get it in the third test, or you may get it in the fourth, or you may get it in the nth time. That nth can be any number, right? A person like me, maybe I'll go with 10 different questions, 10 different hypotheses I'll come to a conclusion. Someone else who is not satisfied with that, they may go with the 20 different questions they may raise, 20 different hypotheses they may make, and finally they can come to the conclusion. Someone who is not interested, who cannot wait for such long time, they conduct just 2 or 3 tests and they can come up with the conclusion. That can be anything, but all these falls under your science. So we are talking about why clinical trials are important. There we understand that we are going to have a scientific research. The scientific research we are going to systematically convert or translate into 3 different ways. That can be either of a preventive charge or it is going to be a treatment drug, or it can be a diagnosis device. Then we come up with a question called, what is the scientific method? Scientific method is all about whenever I give a drug or equipment to you, you are going to test it, you raise a question, you made a hypothesis, get the new test done, get your results, finally analyze it. This is what we are doing here. Any questions? What is called scientific method is? Guys, the reason why we are discussing about this is this may not be straight away affecting your day-to-day work, but if you're appearing for the interviews, right? Someone who really want to test your basic domain skills, domain understanding, then you are going to face these questions. That's the reason I'm starting from the very, 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 very basic. Clear? Any questions? All of you are clear with scientific method? Can I have a quick confirmation?
Yeah, it's clear.
Yes.
Yeah, yeah.
You can sure. You can also respond by using your chat window. So I'm okay with that. I'm happy if you respond orally, but still, if you want to use your chat window also, it is good. All right. Now, three different questions are asked. What are clinical trials? We have a basic clarity now. Number two, why are clinical trials important? Yes, we also discussed that. Number three, how are clinical trials like scientific method, like the scientific method? All these three we already covered. Now, let's move on to the next topic, a very important one. What are the types of clinical trials, right? When I say types, someone needs to define it, right? Everyone have their own definition of what is what, correct? So someone need to standardize it. Someone need to mention that, hey, this is what the definition is. Everyone need to follow it. And that someone is nothing but National Institute of Health. The National Institutes of Health define that there are two types of clinical trials. The number one is going to be, there are not two types, so sorry about it. There are few more. So first we start with the first one, which is nothing but treatment trials. The treatment trials is all about you're going to either test a new treatment, you're going to test the combination of different drugs, or you're going to test a new approach to the surgery or a radiation. Let me give you an example. Um, okay, let's talk about a surgery, right? So appendicitis, one of the most common, I can say that 3 or 4 out of the 10 people generally get appendicitis, right? It is not going to be a disease, but they may undergo this particular procedure. At least one, or I can say 2 out of 10 people will undergo the surgery, right? Initial days, I can say that around 10 years back, when someone have this problem of appendicitis, so on the abdomen, lower abdomen, they are going to make a cut, at least I can say that 10 to 12 centimeters long, right? And then they are going to take away that appendicitis part, right? If any other surgery required, they also do that. And then they are going to put all these stitches and all. We need to take a proper care of those stitches for at least for 10 days or 14 days. And then again, we need to visit the doctor. The doctor is going to check how that surgical area is going to be. And then finally, he or she is going to remove your stitches by cutting them. Correct? And then again, we need to take some more care to get healed with that particular wound. Maybe after 20 days or maybe after 30 days, you'll become normal. Correct? There is a procedure of approximately a month time from the day you undergo the surgery. It will take minimum of 1 month to become a normal guy. Correct? I mean to say that complete healing of the wound. So what's the next stage? Now we are talking about this is the treatment. So what is the improvement to this particular treatment? This is a new approach to a surgery. So we slowly started moving from, instead of having these nylon stitches, right? So why can't we have these dissolvable stitches? those will be automatically dissolved and they will be fallen down. Correct? So what is the impact of it? The impact is going to be less number of stitches are more sufficient. Number two, you don't need to consult a doctor one more time for removing your stitches, right? And the third thing, the total time it will take to get healed is also going to be less. So there is a slight improvement. So to bring back to the normal life, you don't need to wait for 30 days. Let's say that span has been reduced to 20 days. What's the next stage? So we tested one more way of approach, then we move to the laparoscopy. So what we are doing there, there's no need to cut your abdominal, lower abdominal at least 8 centimeters or 9 centimeters. Instead, we just make a small hole. To that particular hole, we do the necessary thing, right? In this way, you don't even need to wait for 20 or 25 days to get this healed and everything. We just make sure that it is going to be more effective and it will take less amount of the time, right? That is nothing but post operation, the post surgery, the amount of time that you need to spend is going to be less. Can I say that this is going to be a new approach? Obviously. All right. So then coming to the treatment of drugs and all, like combination of drugs and all, this is always going to happen, right? So let's say that for someone who come to a doctor, let's say that, hey, I'm having some severe body pains, along with that I have some lower temperature. So before, doctors used to write, a different kind of medicine for the, body pains, and there is going to be a different kind of medicine for your fever, and there is going to be antibiotic just to make sure that all your things under control. Nowadays, instead of having three different drugs, doctor may prescribe a single drug which will be efficient enough because which is having a medical medicine combination which is going to treat all these three problems, right? So treatment trials is all about either finding the new treatment or combine the existing drugs to make sure we are going to produce a most effective drug, or always find new approaches to treat your existing surgery or to improve your surgeries. Correct? Number two, prevention trials. There is always a better way to prevent a disease, right? There is always a scope for that, and we are continuously working on it. So one of the best thing is going to be COVID, right? So it literally shaken the entire world, but now we are somewhat confident enough that, right, at least we can prevent falling sick because of the COVID, because most of us, I can say that 100% of us are already got vaccinated. So vaccine is, like, I can say that one of the preventive measure. So we used to have the vaccine in the childhood itself, maybe that within 2 to 3 years of our age itself, we may have vaccine for polio, we may had a vaccine for pneumonia, we may had vaccine for malaria, correct? There are so many vaccines now, hepatitis, there are a lot of stuff, right? All these are preventive actions to make sure that, to prevent the given disease. In addition to that, I told you there are few more. So what are they? Diagnostic trials, right? Diagnosis is nothing but, let's say that you suddenly feeling even under 24 degrees centigrade of AC, still you are sweating, you are getting sweat, right? So what is the meaning? You may have BP, you may have high blood pressure. That is the reason, though you are under AC, still you are sweating. Still you are sweating. So you need to get diagnosed to see whether you have the blood pressure or not, high blood pressure or not. If not, what exactly the root cause? Someone need to diagnose it. That's where we have all these labs. Correct? Then screening trials. Always find the best way, always try to find the best way to detect or treat the diseases, right? So what is again the example? We can consider COVID itself. COVID can be screened or can be detected in three different ways. Number one, you can simply go with the rapid test. Take a simple, yeah, salvia or anything, just take that, just use your rapid kit and find out whether you're COVID positive or not. There are high chances that there are going to be false positives. right? So what generally we do then, right? Second thing is we go with RT-PCR, correct? So under that, again, when you go with the RT-PCR, it will go to the number of different tests, the different types of screening to identify whether you have any COVID related stuff or not, right? Again, under the RT-PCR, there are different measures. Some labs use, I don't go deeper into that, but each lab will go with different ways of different ways of screening your RT-PCR. Even after RT-PCR, let's say that you got a negative there, but still you are feeling sick. Your doctor is not convinced. What's the next method? He asks you to go with CT scan. You go to the CT scan, make sure your lungs got scanned. So there he will identify what level you have there, what percentage you have there. Based on that particular percentage, the doctor can come to the conclusion. Can I say all these are screening trials, right? So you have a rapid kit test, you have your RT-PCR, you also have your CT scan, right? So you have multiple ways to identify or to detect the best thing, correct? Last one, quality of life trials. Um, in India, this concentration on this particular trials, I can say that little less. But if you see European countries compared to anyone else, European countries, they spend most of their trials on this particular. If you see any new drug that either come from India or it will be somewhere from your Israel or it can be from USA. Most of the drugs I'm talking about, not like entirely, most of them. But if you see European countries, especially countries like Switzerland and all, Austria, Vienna, they and all, they concentrate more on quality of life trials. That is, try to explore more ways to improve the comfort and quality of life of the people, right, who have some chronic illness, who have some chronic illness. So if you see that someone is bedridden, right? The way we treat, the way we live with a bedridden person in India is completely different from the person who bedridden in somewhere in Europe. The equipments, right? The different equipments we have to handle that will be way better compared to India. I'm not criticizing. There are a lot of factors, right? There are a lot of factors why we are not into that particular stage yet. But still, the amount of time, the amount of money that we are putting on this particular trials is less in India compared to the other countries. Clear? So what we discussed here, we discussed about what are the different clinical trials types. What are them? The first one is going to be the very basic one, which is nothing but your treatment trials, correct? So this is nothing but to improve or to have a better treatment. And the second one is going to be your what is the second one? Preventive vaccines. The third one is a diagnosis, and is it third one diagnosis only, right? Yeah, diagnosis. And the fourth one is going to be, sorry, the next one is going to be like once we have treatment related stuff, then we have preventive one. Third one is going to be the diagnostic one. The fourth one is screening, right? And the last one is going to be how to improve the quality of the life, which is nothing but quality of life, right? So these are the various trials that we generally have in under the clinical trials, correct? Now, any questions on this types of clinical trials? All of you are clear with that, right? One main thing we all need to understand is, I have expected this question, but still I haven't get it. I told you that we don't spend much of or we don't do much of clinical trials compared to the other countries. We means India, I'm talking about. The primary reason is clinical trials is nothing but it's more under the research and development. And generally, this research and development, the success rate is always less, a very minimal success rate, right? You need to do a lot of stuff. You need to go with a lot of questions you need to ask, a lot of hypotheses that you need to make, a lot of results you need to analyze to make sure the given drug or the given equipment is working as expected or to meet the demands or not, right? So definitely it is a more time taking process. It won't give you the immediate returns. There are long-term returns to you all the time. So you need a sponsor because you need to invest a lot of money there. Whenever money is there, they require a sponsor. So all these clinical trials are generally funded or sponsored by all your pharmaceutical companies, right? So where these pharmaceutical companies are there, or where exactly these clinical trials will happen? These clinical trials can occur at sites, mean to say, all these trials can carry out at the regular hospital's research and development department, or there are medical universities. So these trials can happen under these medical universities, or some of the specialized doctors. So they won't go with the regular treatments and all. Their primary objective is only to concentrate on the research and development. So those are nothing but the doctor offices. Some of them may happen under the community clinics, right? Or there are few people, they are dedicatedly working as a clinical trial contractors. In those offices also it can happen. So what is more important to us here? The most important thing here is going to be where these clinical trials happens. The clinical trials happens at any place, say it can be a regular hospital research and development department, or it can be under the medical university, or it can be under the supervisor or a specialized doctor office. It can be a community clinic, or it can be at the offices of your clinical trial contractors. All right. So before we go more detail into this, let's say that what are the various clinical trials we have? Clinical trials generally happens in the phases, right? So which is nothing but the very first step is nothing but preclinical trial. Preclinical trial. It is the first stage. What exactly the preclinical trial is? You're finding a new drug, or you're going to bring a new medical device, or you're coming up with a new procedure that we haven't done on any human being before. So before you do this particular trial or testing on the human beings, you generally do it on animals. Remember this. All the preclinical trials, the first trial always happens on the animals. Why do so? Why we need to do that? To understand the mechanisms of the action, to determine how well the treatment is working, and to see the main thing, is it safe to test on humans or not? Of course, we are not so generous. Of course, we are not doing the right thing towards the animals. That are all the second story. But the first thing that we need to understand is the initial stage for your clinical trials is preclinical trial. The preclinical trial must be carried only on the animals. Why we are doing that is primarily to understand how safe it is, whether it is working or not, and what exactly the mechanism of that particular action. All right. So that is the most important thing. Okay. Let's take a small pause here and try to understand this. Who sponsors the clinical trials? We understand this. Why it is important to test the drugs? We understand this. And why it is important to test the drugs on the animals rather than the human beings in the first stage? We also understand. All of you are clear up to this? If I summarize what we discussed up to this, We understand what is called clinical trials, the first thing, and then we understand the types of the clinical trials, and then we started with the phases of the clinical trial. In that, we just started with the preclinical trial. It is generally carried out on the animals. Questions? Any questions up to this? All of you are clear? All right. Questions? Fine. This is the another important thing. So let's say the preclinical trial is done. Now we are entering into the phase one. The phase one is always happen. It always going to do only on a smaller group of people, and the range can be from 20 to 80. That is nothing but the number of people who will be part of this particular phase one, or maybe minimal 20 to 80. If you remember, when we go with this COVID vaccine, right? So which one it is? I think Bharat Medicals, right? So only within the organization, whoever are part of this particular R&D, only those guys have taken that particular vaccine first, right? A very minimal number of people. Similarly, so always the phase one is only on a smaller group of people, and it should not exceed 80. Sometimes it can be even lower, under 20, even 10 people will be part of, but always make sure that it is going to be a very small range, right? The main purpose of this phase one is, again, at every point of time, we will be talking about the safety. That is the main thing, safety. So first we are going to evaluate the safety. We know that it is safe enough on the animals. That is the reason we are taking this particular medicine, or we are using this particular device. But again, we need to check the safety of the human being. We need to evaluate that. Number two, at the animals level, we don't look at the side effects much, because side effects are not going to be considered as a major aspect when it comes to the animals. But when it comes to the human beings, we start evaluating the side effects as well, right? What are going to be the side effects? And then main thing, when I say that, hey, I'm just going with the clinical trials on a smaller group, from where these people will come? How do you find them, right? Most of the time they are going to be the volunteers. Most of the time. Sometimes they will get paid as well, right? Would you volunteer to be a subject in a clinical trial? What if it mean to you receive a treatment for a disease that wasn't available to anyone else? Very, very important thing. Most of the people, there are some of the contractors, there are some of the people whose main thing is, whose major income is to be part of these clinical trials. Some of them will do generously, but most of them will get the money for it, and that's the reason they have these trials. So the phase one, smaller group, maximum ranging from 20 to 80. The main intention is going to evaluate the safety of the particular drug, and number two is to evaluate your side effects. Phase number two. Now we are going to expand the range, right? The drug is working fine, or at least it is giving the good results on the smaller group, right? So why can't we expand the group, right? But while expanding the group, we consider a lot of other factors as well. We go with the different age groups, we go with the different genders, we go with the different people from the different continents, to make sure it is working for all of them. Again, safety is the most important thing here. Next to the safety, we are going to test here the effectiveness, the efficiency we are going to determine here, because we know that it is safe for the animals, it is safe for the smaller group of the people who are human beings already. But still, I consider the evaluation. Now I'm going to extend my evaluation now to the different group of people. Those groups can be based on the age, those groups can be based on the gender, those groups can be based on anything else. But again, we're expanding the range. That is nothing but your phase 2. Now, the phase 3, right? So when it comes to the phase 3, we are going to expand the range to a lot. I can say that almost it is going to be 10 to 15 times of, minimum 10 to 15 times of your phase 2, right? Which we expand our range from 300 to 3,000 people. Here, the major objective is to find the effectiveness, right? I'm just checking the effectiveness compared with standard or equivalent treatments. What do you mean by the standard or equivalent treatment? There is already an existing treatment available. There is already one drug available to cure this particular disease in the market already. People are using it, right? So when I compare with that particular drug, how efficient my new drug is. When I compare with the existing drug, do I have any side effects? When I compare with that, let's say when I use the existing drug, I'm having some dizziness. The dizziness is the side effect of having the particular drug. When I go with the new drug that I'm developing now, even here, do I have the dizziness? If the dizziness is not there, then we say that compared to the previous drug, our drug is good because we don't have side effects now. Let's say that dizziness is not there, but it is causing some swelling, or it is making it is producing some rashes, right? Or some other treatments, then I need to take it care of. Something is not good with my drug. So this in phase 3, we extend the group to a larger number. Then we're talking about the effectiveness of this particular drug, and we are also talking about the side effects compared to the existing drug, which is already present in the market, right? So we already have a benchmark. We need to overcome that, or at least we need to match that. Clear? All of you are clear up to this? Right? There are few more trials here, but I don't go into that particular one. I'll give a pass here, and I'll tell now where SAS is required and why SAS is required in this entire clinical trials. All right. Let's say that. So we have done a lot of research and development. Let's say this is company A. After a lot of research and development, we come up with a drug A. So this drug, I did it on the animals, that is preclinical, phase 1, small group of people, phase 2, extended group of people, phase 3, a larger group. All the 3 phases are done. And I'm sure that this drug is really doing the wonders. Can I release in the market directly? Yes or no? Guys, can I release it in the
No,
market directly? Okay, what needs
no.
to be done? What needs to be done? We need someone's approval. Am I correct? All we need is an approval. To approve, someone needs to approve it, and that someone is nothing but your FDA. Right? We'll talk about all the acronyms later, not an issue. Right? Just understand that there is some monitoring unit at the top in each country. That particular country's that monitoring unit has to approve this. Correct? Only after the approval, I can release this particular drug into the market. That I can be done. But before dropping this particular drug into the market, I need FDA's approval. I'm talking about the US market. I'm talking about FDA. Okay. Let's say that someone has sent this particular drug A to the FDA. Similarly, there is another company. Let's say consider COVID itself. So what do we have? We have Covaxin, we have Covishield, right? And we also have the, what is the other one? Sputnik, right? And the new drugs are keep on coming, new vaccines are keep on coming. So let's say that there is a company A and company B. So drug A and drug B, there are 2 drugs which are produced by 2 different companies. Both of them are going to cure the same disease. Let's say there's a third company as well. So how do when I receive F drug A, drug B, drug C, all the three drugs are received to the are came for the FDA's approval, right? So every day hundreds of drugs will come for the approval in the form of a report, correct? I'm going to generate one report, and that report I'm going to use it for the evaluation purpose. Let's say I sent it in an Excel. Means I belongs to the company A. I sent all the details related to the drug A in Excel sheet. There is a company B, they send it in some XML format. Company C, they send it in a HTML format. All of them are going to have the very good information. The information is really good, right? They have all the information which are related to that particular drug. But how easy for me or how difficult for me being under FDA to evaluate them? Because they're not following any same format. Each individual is sending in a different format. So can I evaluate them on regular basis? Difficult, right? I don't have a standard. Until and unless I have a standard, right, it becomes a headache for me to evaluate them. So there need to be a standard. There need to be a standard guidelines, right? Let's say that all of them are sending all these in a Excel format. Let's say that. I'm just telling you. Again, in the Excel, when being a drug A, all the drug A details, let's say that I'm saying that I'm using the term called gender to represent male and female, right? I'm saying that gender is equal to NALE, FEMALE. I'm writing in a full form. When I search for gender in the other company document, Excel sheet, I don't find gender there. Instead, they mention it as sex. So both of them are representing the same thing, but my representation is gender, other company's representation is sex. So there is difference in variable indication. There is a difference in how we are sharing the information. So I have standardized the format, but I also need to standardize the parameters. Each parameter must be in a standard way, correct? Let's say that we have given the headers. Hey, anyone want to represent male or female? You should be use the name called gender only. The parameter name must be gender. Okay, fine, done. Again, when I see that company A has given gender as M and F. Company B has given the details as male and female. Company C has given the details as 0 and 1. 0 means males, 1 means females, right? According to them, each of them are correct. But again, when it comes to the FDA, when they are evaluating, should they need to consider FEMALE or should they need to consider F or they should need to consider 1? All three are three different values for the same parameter. So again, we need to come to a conclusion that we also need to standardize the values. It's not just about the parameters. It's not just about the file format. It is also about the standardization of values. Gender means everyone has to follow either F or M. That's it. Dot. Clear? So someone has to give this standard implementation guide. Hey, anyone want to generate this particular report and give it to me? You need to give me in this particular standard format only. That format is nothing but your SDTM. So what they said that they have given some standard implementation guide. This is implementation guide. Everyone has to follow this only by using this particular guidelines. You need to send the report to me to produce that particular report, right? By having all the SDTM implementation guidelines based on that, I want to produce a report. To produce that particular report, we are using SAS. Because to produce this, we need to work on so many data sets. There are lots and lots of data sets that are available. I need to do a lot of analysis. I need to do a lot of reporting. To do all this, I need a technology, and that technology is nothing but your SAS. So the role of SAS here is By strictly following your implementation guide, generate a report which we will send it to the FDA. FDA is going to evaluate that particular one. Either they are going to approve it or they are going to. Do you understand now where we are and what we are doing? Can I have a quick confirmation from everyone? All right. So here I just have given the same details. You can see that SDTM stands for Standard Data Tabulation Model. We're also going to discuss about ADAM, which is nothing but Analysis Data Model, the next level of your SDTM. And then we also talk about TLF, which are nothing but tables or listing figures. All these three are going to be covered under your clinical trials. Again, you can see phase 1, phase 2, phase 3, all these things. I just told you drug A, drug B, drug C, 4 different companies, the same thing I just mentioned here. So now to make sure I'm going to standardize it, I'm going to use the standard implementation guide, right? So who is going to do this? Who is going to monitor this? Who provide the standard implementation guide? CDIS, Clinical Data Interchange and Standard Consortium. These are the guys. CDIS are, even if you Google it, you're going to get it. A simple thing, cdis.org. This is the organization who are going to use or who are going to maintain these standards, right? These are the ones who is implementing, who is monitoring, who is doing all these SDTM things, standard, what is this? Standard data tabulation model. This implementation guide is actually maintaining, monitoring, producing, all these things will be taken care by your leaders. All right. Now, so we know where SAS fit into, what is called your SDTM is. We haven't deal too much about the SDTM because we are going to start discussing about that. Today's discussion is only to understand what is called clinical trials, where SAS fit into, and what is called your SDTM on a very basic or low level. From tomorrow onwards, what we are going to do is we start discussing about SDTM. The current version is going to be 3.2. That is the latest guidelines. Every time there is any changes in the guideline, we are going to get a new version. The latest implementation guide is 3.2. Based on the 3.2, we are going to have a discussion, right? Every day we are going to discuss about what is this SDTM, what are the various domains we have. For each domain, we are going to understand all the variables. For that particular one, we are going to start parallelly, we are going to working on the practicals. All right. Remember, guys, clinical SAS.
Before SDTM, you are not going to explain about raw data. Uh, quick
Raw data
explanation
is
about
nothing.
raw data.
Raw data is all about your data, right? What data sets that we are using and all. Is that what you are talking about? Raw data is,
Yes.
see, the raw data I start introduced to you when I start giving the practicals. At least one domain I'll explain first. But that particular domain, what are the data sets that are required? That raw data I'm going to explain, and then I can start giving you the practical stuff on that. All right. So without knowing what is the
Okay.
standard implementation, why do we need to understand about the raw data? Raw data is all about the trials that are already conducted and the recording, the readings are recorded, right? So those we go for each of the different domain, we go separate. Questions. So there are certain questions in the chat box. What days we are going to have the class? Generally, it is Monday to Friday, 1.5 hours. That is how it is. Maybe I'll see to that and I'll confirm on it. But now just consider that it is going to be a daily class, 1 hour class. Maybe next week onwards, I'll tell you if either it is going to be an alternate day for 1.5 hours or it is going to be every day 1 hour. I need to confirm that. I'll check and I'll confirm. Okay. All right. Any other questions here? All right. If there are any questions, I'm happy to answer. If there are no questions, we can wind up the session for today. Vamsi Krishna, videos, you can just have a word with Balisar. Balisar can help you with the videos. The recording will be available after 2 hours from now, so he will upload into the respective drives. You can get the confirmation from Balisar. Yes, class will be on the same time. It can be either 8 to 9 or 8 to 9:30. That alone we need to discuss. I'll give you the confirmation for this week. Tomorrow and day after tomorrow also, it is going to be 8 to 9, 8 p.m. to 9 p.m. IST. All right. Thank you, everyone. See you tomorrow. Take care. Bye.